Hi everyone, this is our lecture on peripheral B-cell responses. Unfortunately, I can't be on campus today, so I'm providing this video, but this video is for the lecture that would have happened on November 6th, 2023. Previously in the course, we have been thinking about T-cell responses in the periphery. So remember, we followed our T-cells through T-cell development. The T-cells completed their development in the thymus and then left that primary lymphoid organ in order to go to secondary lymphoid organs like the lymph node or spleen, where they were able to respond to a microbe and carry out their effector function. We're now going to see B cells kind of doing the same thing. So when we last saw our B cells, they were finishing B cell development in the bone marrow, and they were immature B cells. They underwent some central tolerance testing, and then if they made it through that, they left the bone marrow as mature B cells. But then we didn't really follow up with the rest of the details of what those B cells did after leaving the bone marrow. Now we're going to be looking at what those B cells are doing in the periphery. Again, looking for antigen and potentially doing their effector functions. And we're going to be in those same secondary lymphoid organs that we have been in, the lymph node, the spleen, and some others like the Peyer's patches that you see here. When we talked about this process with T cells, one of the first things that we had to talk about was the way that we got antigen presenting cells like dendritic cells and T cells into the secondary lymphoid organ like the lymph node in order for us to make that T cell response, for those T cells to be activated and make their response. We're now again looking at the same thing in parallel with B cells. As you'll see in the next couple of slides, T cells are going to be particularly important in this process. This is actually why I had to wait until we talked about T cells before I could tell you about peripheral B cell responses. And in the case of B cells, secondary lymphoid organs will collect antigen, specifically free antigen. Remember that B cell receptors and antibodies bind to antigen in its native 3D form, not denatured on MHC like T cells do. So we need that native antigen, which comes into a secondary lymphoid organ. We need some T cells and we need some B cells. All of those things will end up in the secondary lymphoid organ. The T cells and B cells can come in through the blood vessels, potentially through an HEV like we saw previously, while the antigen is largely going to come in through the lymphatic vessels. And you can see on the right, T cells coming in through the HEV, going to the T cell zone and doing some of the exact details that we thought about earlier with T cells becoming activated by a dendritic cell, while the B cells will also come in. They will go to a B cell zone and they will start doing some of the events that we're going to talk about today. When one thinks about B cell responses, there are lots and lots of different types of B cell responses, just like we saw with T cells. One way that we can classify B cell responses is as either T-independent or T-dependent B cell responses. I'm going to tell you a little bit about both T-dependent and T-independent responses here, but what I will also tell you is that big picture, largely for the rest of this lecture and for the rest of this course, we're going to be thinking about B cell activation as something that's happening in a T cell dependent way. So we'll be thinking about the T dependent activation, but it is important for you to know that it is also possible for B cells to be activated in a T independent way. If you remember when we thought about how we could activate a T cell, we talked about the importance of signal 1, a signal coming through the T cell receptor, and signal 2 coming through our co-stimulatory molecule, which in this case was CD28 on the T cell. With B cells, we sort of have the same kind of thing happening where we need to have multiple signals. Here you can see a signal 1, a signal 2, and a signal 3. And the big difference between T-cell dependent and T-cell independent B-cell activation is exactly where the signal 2 is coming from. So what you can notice here at the top is that in a T-dependent situation, signal 2 is actually coming from the T-cell. We can have signal 2 come from some other places like PRRs or like some additional receptors like a complement receptor, and they can turn on a B cell in a T cell independent way. But the outcome of that B cell activation is different. So yes, we can turn on a B cell. You couldn't see me doing air quotes when I said that, but we're sort of turning on a B cell in all ways. But we're turning that B cell on in a particularly good way with the T cell dependent activation, where we're getting this signal 2 from T cells. And so specifically, when we have T cell dependent activation, then we can get this process of class switching. Then we can get this process of affinity maturation. Then we can get this process of immune memory happening. Those types of things are not things that happen if the B cell was activated independently of a T cell. And so that's one thing to be aware of. We are going to largely be talking about some of these types of effector functions that all require T-dependent activation. One other thing that you can notice on this slide is that this table reminds us that T-dependent antigens are only protein antigens, while T-independent antigens can be other types. This is because in order to have good T-cell responses, we had to have a protein antigen that could be bound to MHC. So we can only get a T-cell dependent response if we have a protein antigen. And you can see, again, the big picture overview of what happens when we have a T-cell dependent type of B-cell activation and when we have T-cell dependent B-cell responses. So you can see our T-cell here, which is shown in blue, interacting with our B-cell that is shown in yellow. You can see that on the B-cell, there's a protein called CD40. CD40 is the signal 2 or the co-stimulatory molecule on the B cell. So it, in this case, is sort of analogous to what CD28 was doing on our T cell. So CD40 is that signal 2 protein for the B cell. It is binding to a protein called CD40 ligand. CD40 ligand is on the T cell. You can think of it as being analogous to B7 that was on the antigen presenting cell from what we saw in the case of T cell activation. And if a B cell is activated and gets this signal 2 through CD40, that B cell will proliferate, that B cell will differentiate, and will potentially become either a memory cell or a plasma cell that have slightly different functions that we'll be talking about later this week. That B cell can also undergo affinity maturation or somatic hypermutation, and we'll talk about that in a bit, where that B cell actually changes its variable region in order to bind antigen better. And that B cell can also undergo class switch and switch from perhaps IgM to maybe IgG and make a better response for a particular type of microbe. So in order for this process to happen, the first thing that needs to occur is that the B cell needs to encounter antigen. And just like we saw with the case of T cell activation, this first encounter with antigen is happening in our secondary lymphoid organ, specifically in the lymph node. And so here we can see that we're going to have some antigen. Here it's shown as a red pill-shaped thing that's just hanging out in the lymph node and will bind to the B cell receptor. And when that happens, the B cell will actually get a signal to internalize that B cell receptor that has antigen bound. So you can see that happening here. That B cell receptor will actually get recycled back up to the surface to try to pick up some more antigen. And that antigen in the B cell will be processed and presented on MHC class 2. Remember that B cells were one of our professional antigen presenting cells that had the ability to present on MHC class 2, even though they are not cells that do phagocytosis. Instead, they do receptor-mediated endocytosis like you see here. This process starts to activate the B cell, but does not fully activate the B cell. And you can start to see this at the bottom as well. So at the beginning, we might have a resting B cell. If that resting B cell encounters antigen, that resting B cell will internalize that antigen, and it will start making much more MHC class 2 on its surface, where it will be presenting different peptides. It will also start making lots of other types of proteins that can activate good immune responses. But one of the other things that B cell will do is that B cell will start to move around the lymph node. Our B cell originally comes into the lymph node through a blood vessel, particularly a high endothelial venule, an HEV, just like we saw with T cells. And because of chemokines that are in the lymph node, that B cell will move to an area that is best described as the B cell area. And that B cell area will be a place where there will be a lot of antigens present. Some of the antigen will be free, some of the antigen may be in another form that we'll talk about in a little bit on something called the FDC. And so here you're seeing on the left these naive B cells first going to encounter antigen upon initially coming into the secondary lymphoid organ like the lymph node. And then on the right, you can see those same B cells that have started the activation process that we saw on that previous slide. So they've gotten a signal through their B cell receptor. They've gotten sort of their signal 1, but they haven't fully been activated yet. And what you'll notice is that those B cells move, and they move to this area of the lymph node that's sort of on the boundary between the B cell zone and the T cell zone. So you can see that movement of those B cells in this image. Once the B cells have moved to the boundary between the B cell zone and the T cell zone, now T cells and B cells can interact. We're sort of at the boundary of their two areas. And now T cells are going to interact with B cells in the lymph node or other secondary lymphoid organ. Specifically, these will be CD4 positive T cells, and they will be able to provide T cell help to the B cell. And specifically, they will do that by making CD40 ligand that will now bind to CD40 on the B cell and give that B cell its signal 2, so that B cell can now fully be activated. And so this is happening at that boundary between the B cell and the T cell zone. Originally, we just sort of thought that these were helper CD4 T cells. We now know that specifically the CD4 T cells that are doing this job are the T follicular helper cells. And so you can see that our naive CD4 T cell will have to first get activated in the lymph node with its antigen and with specific polarizing cytokines that will make that T cell turn on BCL6 and start making cytokines like IL-21, and that will allow this T cell to start helping B cells. Once that T cell is activated as a T follicular helper cell, it will also start making CD40 ligand. And so you can see this process happening here. Our B cell will encounter some free antigen and internalize that free antigen. That B cell will then process and present that antigen on class 2. And what you can see is that it's actually presenting that same antigen to this T follicular helper cell on its MHC class 2 to allow it to interact with that T follicular helper cell. So this B cell presentation of antigen is key in getting the B cell, shall we say, additional attention or additional pats on the head now from the T follicular helper cell so that that B cell can get its full activation. And when that T cell receives that MHC class 2 presented peptide from the B cell, now you can see that T follicular helper cell is going to start making CD40 ligand. It's going to start activating, making CD40 ligand to bind CD40 on the B cell and turn that B cell on. And now we're going to get full B cell activation. And the cytokines that are produced by the T follicular helper cells are also going to be really key here, sort of as a signal 3, just like we saw with T cells. Here you can see this same process, but now thought about in terms of the spatial places where we see it. So first, the T cells that are naive T cells come into the lymph node. They interact with a dendritic cell that's presenting antigen, and they become activated, and they may receive the signals from that dendritic cell so that they can be polarized into T follicular helper cells. While that's happening, our B cells will also come into the lymph node, and they will go to the B cell zone where they'll interact with antigen, and they will then start to be activated, and they will move to this boundary region between the B cell zone and the T cell zone. When our T follicular helper cells are fully activated, they will also move to the boundary zone. And now you can see those T cells and B cells interacting with one another. So we've got one T follicular helper cell and one B cell that started its activation process, making a pair. And that T cell will be able to, just like we saw on the previous slide, provide signals through CD40 and through cytokines to fully activate that B cell. Once that B cell gets this kind of signal, that B cell is going to go on to another area of the secondary lymphoid organ or the lymph node that's known as the germinal center. So once a B cell has sort of had this interaction with a T cell, it's now ready to go to a germinal center. You can see this exact same thing happening here as well. So you can see these B cells eventually moving to these areas called the germinal center. If we look on the right-hand side, you can see some images of lymph nodes and different times after antigen. B cells are generally in blue, although B cells specific for our antigen of interest are going to be in green. And T cells specific for our antigen of interest are in red. So you can see that when we don't yet have the antigen, when the mouse is naive, we don't really have a huge number of those cells in the lymph node. We can see once the antigen gets there, we get more and more of those cells. They're in two separate areas. Those cells then kind of start to move close together. And then the B cells will end up in this area here that we can see as sort of a very defined area that's known as the germinal center. Sometimes people want to define germinal centers, and there's some weird debates about how you define a germinal center. Sometimes you only can say, well, a germinal center is the place the B cells moved to. It became a germinal center when the B cells picked to move there. It wasn't like special before, or it maybe was special before. The one thing that we need to know that does exist in a germinal center is there is a particular cell type called an FDC that must be present in the germinal center. So those B cells will move to a place where there are FDCs and perhaps then just start doing their stuff to set up a germinal center there. The germinal center has different zones that we're going to see in this process, and you can already start to see them. The area kind of in The sort of B cell-ish zone that isn't germinal center is usually called the follicle. And then in the germinal center, which is in these darker colors of blue, we often will talk about areas called the dark zone and the light zone. And you'll see them a little bit more. But I want you to think a little bit about some things we've talked about with previous immune organs already. If you remember, when we talked about the thymus, we looked a little bit at the cortex and the medulla, and we looked at how those two parts of the thymus looked under a microscope. And one of them was a lot darker than the other under a microscope. If you remember, we talked about the fact that this was because one of those areas was an area where many cells underwent proliferation. It was a place where cells divided, and so the cells got really crowded there. That made it hard for light to go through, and it made that area dark. While other areas were areas where the cells were not actively dividing, and in some cases actually undergoing cell death, so the cells were not as actively as dense there, light could go through easier, and that made that area light. Well, the two parts of the germinal center are known as the dark zone and the light zone. So hopefully you can notice already that perhaps one of those zones might have something to do with proliferation. It might be the dark zone, as opposed to the light zone. So you can see a lot of similar themes to things we've already seen in some of our previous lectures. So here you can see free antigen potentially entering the lymph nodes through the lymphatics. And in some of the previous slides, I've just sort of said the free antigen gets into the lymph node. The free antigen will bind to the B cell receptor. The free antigen ends up in the B cell zone. And all of those things are true. Sometimes that free antigen just stays as free antigen, which you can see here just as these random little brown, I don't even know what that shape's called, these random little brown shapes throughout the lymph node. But sometimes that free antigen can enter the lymph node and bind to a particular type of cell called a follicular dendritic cell. Follicular dendritic cells you can see here. They are also abbreviated as FDC, and they have been labeled as FDCs on some of the previous slides. Follicular dendritic cells are a type of cell that frequently confound immunology students. I think they're every immunology student's least favorite type of cell. So I apologize for them now. I promise I'm not the one who named them. One thing that's nice about a follicular dendritic cell is we do know where it lives. It lives in an area of the secondary lymphoid organ like the lymph node that's the follicle, so that part's good. But the thing that's less good about a follicular dendritic cell is that follicular dendritic cells are not dendritic cells. Somebody a long time ago looked at them under a microscope and thought that they looked like dendritic cells and thought that they were dendritic cells, but now we know they're actually totally and completely not dendritic cells. That means they do not do phagocytosis. They do not present antigen on MHC class 2. When I talk about follicular dendritic cells, I really don't ever use the word present antigen. Yeah, they are nucleated cells. Yeah, they could have stuff on class 1. But we're really not thinking about class 1 here. We're thinking about class 2. And they're not doing phagocytosis. They are not doing MHC class 2 presentation. And so we're going to talk about follicular dendritic cells and their interactions with antigen, but it is not antigen presentation in the way that we talked about antigen presentation earlier in the semester. Instead, I like to think of follicular dendritic cells as being a little bit like flypaper. That really sticky paper that you might hang up to try to get stuff to fly into it. The idea that it's just really sticky and has stuff stick to it. You can see a follicular dendritic cell on the right hand of this slide, and you can see It has all sorts of nice long processes. You can see how somebody might have originally thought that that was a dendritic cell. Um, the good thing about that shape is that gives the cell a lot of surface area to be sticky. Because remember, it's having a sticky surface is super important here. And those follicular dendritic cells, you can see a follicular dendritic cell here in blue on the left, just have all sorts of different types of receptors on them that allow them to have antigens stick to them. And so a follicular dendritic cell may have something like a complement receptor. So any antigen that is bound to complement will get stuck on follicular dendritic cells. Follicular dendritic cells have a number of other types of receptors, and you can just basically imagine a follicular dendritic cell as just this nice surface that all the antigens are getting stuck to. And so it's sort of like a display case of all the free antigen that could be around. It's not processed, it's not an MHC, it's just all of the different antigens that are in that region of the body stuck all over the surface of this cell. Questions about things like how long antigen remains on follicular dendritic cells, and some details of how that impacts things like virus replication are really interesting and are things that immunologists argue about. But basically, you can imagine the follicular dendritic cells are just this little area or this sort of cell type that lets every possible antigen stick to it and actually stick to it for a decently long time. And so provides a source of antigens, foreign antigens really, for B cells to go through some of these activation processes with. Sometimes they will be part of that earlier activation that we were talking about, but they're also important locations around which those B cells that are going through this activation process will be able to form a germinal center. And so here you can see a germinal center. The germinal center is this little yellow area, little yellow circle within the B cell zone. You can see it's kind of next to the T cell zone because T cells are going to be pretty important for this process. And we have a dark zone and a light zone in the germinal center. And you can see that with a few different types of microscopy staining. You can also notice that we have slightly different cell types between the light zone and the dark zone. So we tend to have some more T cells in the dark zone, but we'll get to that in a second. You can also notice that our follicular dendritic cells are largely found in the light zone, not so much in the dark zone. The dark zone is really where our developing B cells, as you'll see, are going to proliferate. So let's think about what's going on with our B cell. Remember that our naive B cell has come into the secondary lymphoid organ. As a naive B cell, it's looking for its antigen. And that naive B cell perhaps will find its antigen somewhere in that secondary lymphoid organ and get sort of an initial activating signal, but needs sort of that second signal, need a signal 2. And so that cell might interact with a T cell. This is T-dependent activation. And that cell is now going to sort of have gotten its pats on the head and know that it's time to do things like proliferate and differentiate and start to gain its effector function. So this is just like kind of the T cell when it sort of is like, oh, I'm not supposed to be a naive CD4 cell anymore. I'm supposed to be a Th1. Or, oh, I'm not supposed to be a naive CD8. I'm supposed to be a CTL. That process, the B cell, once it has interacted with a CD4 T follicular helper cell, is now like, okay, I got to go through my like B cell growing up process. And when it does that, it moves to this area called the germinal center, and it goes through a set of steps that are generally referred to as the germinal center reaction. And the germinal center reaction is a really, really cool aspect of B cell biology, where we basically get to see evolution of B cells in action, kind of in real time in a person. I will also tell you that, at least, maybe this is just me, but with some of the things that we have learned during the last couple of years with the COVID pandemic and looking at some details with B cell responses and vaccines, I feel like this is the area where I personally have learned a lot of really cool new immunology in the past couple of years. So I think that there are a lot of interesting things that are coming out in this process. So again, we've got our B cell. It's got its pat on the head. It's told, okay, you need to go grow up and learn how to become a B cell. And the first thing that B cell is going to do is that B cell is going to move to this area, the germinal center, and that B cell is going to do some proliferation. So that B cell is going to make some more copies of itself. It got its pat on the head. We need more copies of that B cell. It's good. But that B cell is also going to do another process at this point. And that other process is called somatic hypermutation, sometimes abbreviated as SHM. Somatic is a word that means body cells. So not like a germ cell or something like that, just a random cell of the body. And usually cells of your body, somatic cells, are not supposed to mutate very much. We don't want to have mutation. We certainly don't want to turn on mutation in a somatic cell. But in this case, this B cell is going to turn on an ability to have mutations. It's not even going to be a couple mutations, a small number. It's a lot. It's hyper mutation. So our B cell is going to proliferate and divide in the dark zone, make more copies of itself. And that B cell is also going to undergo this process of somatic hypermutation. And we can see some of the details of somatic hypermutation here. So we're going to be getting a lot of mutations in this B cell, and we're not just going to get them randomly throughout the genome of the B cell. Instead, we're getting specific point mutations. So we're getting, one base pair changed to another kind of base pair, specifically in the variable region of the genes that make the antibody. This could be the heavy chain or this could be the light chain. And so you can see in this version on the right, we have the variable and the constant region encoding our antibody, and we might pick up some mutations in the variable region, different point mutations. So we've got lots of changes to the DNA, lots of mutations. This is also shown on the left, where you can see a graph of the frequency of mutations along different parts of the antibody gene, and you can see they're really all focused largely on kind of the V and J, or the variable region, and there really are almost no mutations outside of the V and J region. Because we're making changes to the DNA here, the B cell can't go back and fix it. This is sort of an irreversible change, but changes to DNA will lead to changes in the protein that the DNA encodes. And again, these are point mutations. So probably what's going to happen is we're going to change one amino acid to another in a few different locations of the protein. Specifically, we're impacting the variable region of the heavy and light chain of the antibody. And as a result, we're going to change the ability of that antibody to bind antigen. And so we might go from this binding that's sort of, as this says, low affinity, or as I might say, meh, and we might end up with a protein that binds now really, really well to antigen. We get this even better binding because we've changed some amino acids. and that's because of the changes in the DNA. That change is really a change in the affinity of the antibody for antigen. And so sometimes we call this affinity maturation. The affinity for antigen has changed, gotten better, or matured. Remember that all of this is happening right around the same time that our activated B cell is undergoing some rounds of proliferation. So our activated B cell is making a whole bunch of copies of itself, like, hey, B cell, you did a good job. You're getting your pat on the head, and maybe it will make 100 progeny. Each of those progeny will pick up different mutations in their variable region genes. So now we have this one good B cell has made 100 progeny, each of whom have slightly different mutations. And the goal is that we can perhaps get some nice higher affinity for that antigen. You can see all of this here on the left as well. And so here you can see our B cell. That B cell is going to get its help from a T follicular helper cell. And that B cell will make lots of progeny, including, and will do this process of somatic hypermutation. That's awesome. But this is a random mutation process of the variable region. It's possible that the B cell will make changes to the DNA that result in changes to the protein that make even better binding to antigen. That would be amazing. That's what we really want to have happen. It's also possible that that B cell could make mutations that will not really change the protein at all and have no impact on the affinity. It's also possible that that B cell could pick up mutations in the variable region that completely destroy the variable region and make that variable region useless. It used to be a good variable region, and it's now been made into a terrible one. And so among the, say, 100 progeny that our B cell might make in this process, it's going to make a range of different changes to that variable region, some of which are going to be way better, some of which are going to be way worse, some of which are going to be sort of equal. Now what each of those progeny B cells will do is those progeny B cells will actually test to see whether or not they can get a signal. Can they still get a signal from antigen? So they will go to a follicular dendritic cell and try to interact with a follicular dendritic cell and a T follicular helper cell to get more signal, to get another pat on the head to tell them that they've done that mutation process correctly. If we've made a better affinity antibody, then now this B cell is going to get some really nice signals, and that B cell might divide again and sort of keep going in this process. But if we've destroyed the variable region, that B cell is now not able to interact with the follicular dendritic cell and the T follicular helper cell. It can't get a signal, no pat on the head, and so it's going to die because it cannot get any signal. And so what will happen is our B cell will come into the germinal center. It will proliferate a lot. That's why this zone is the dark zone, because it's super full of cells. And all of those cells are also going to undergo somatic hypermutation. The cells that are a product of this will now move into the light zone, where they will test their newly made B cell receptor to see if they can bind to antigen on a follicular dendritic cell and whether they can bind to a helper T cell. And hopefully, if this worked, if they got a better response, they're going to get their pat on the head, and they might actually move back into the dark zone, proliferate again, and go through this process again. And they could sort of circle through mutating and testing multiple times. Exactly what makes them stop is not really super clear. But what you can imagine is that this really allows for sort of evolution of the B cell response to become a B cell response that binds even better to antigen than it did before. A lot of this is really a competitive response. So it's about which B cells can sort of win out the competition for antigen and for the T follicular helper cell. And that competition is really going to be won by those with the highest affinity. They're going to, get all of the binding sites and get all the attention. And so those B cells are going to go through this process. At some point in this process, those B cells will also do this other thing called class switch recombination, as well as differentiate into our later effector types of B cells. And so you can see sort of the overall results of the germinal center reaction here. So we start with our kind of naive B cell. It interacts with antigen to start to become activated. It also has to interact with a helper T cell. It will eventually expand. It will undergo somatic hypermutation so that its affinity will mature, but it also may be able to further become a cell that is really, really well specialized to make huge amounts of antigen. That cell is called a plasma cell, and that plasma cell makes just an enormous amount of antigen and becomes very, very highly antibody, and becomes very, very highly specialized as an antibody factory. So it does high-level antibody secretion. So if you, for example, are trying to induce a good antibody response, really what you want is this whole process to happen so that you can get some plasma cells that can make you boatloads of antibody. We can also get that B cell to become a memory B cell that may live for a long time. You can imagine that's also pretty important with, say, a vaccine response. And we'll get our B cell to do isotype switching. So that B cell at the beginning, remember, always makes IgM. We saw that during B cell development. And now that B cell is going to maybe switch to make a different isotype, perhaps IgG. And so here you can see CSR also happening as a result of the germinal center reaction. That's class switch recombination. And so that B cell is going to do its class switching, or it may become a memory cell or a plasma cell. And so all of these things are end results of the germinal center reaction, as well as affinity maturation, that improvement in affinity. So here you can see sort of the very big picture overview for class switching. So we saw the 5 different isotypes of antibodies earlier in the semester. You'll remember that I talked about MDs, give everyone apples, for IgM, IgD, IgG, IgE, and IgA. So you can see those different isotypes here at the top. And you remember that they were encoded by these different constant regions that were downstream of the VDJ segments on chromosome 14. You can see those regions here, M and D, being the ones that were really close to the VDJ region. You can see they're only 6.5 and then 4.5 kb away, so they're pretty close. And then there is this giant stretch of DNA in between, 55 kb, before you get to all the rest of these constant regions. And we talked about how, if you remember, that early B cell might be IgM positive and IgD positive. It can make both of them. That's because the polymerase is able to copy this M and D constant region, but it gets too tired. It can't make it this whole 55 kb, and so it can't make all the other ones. So at the beginning, we're really just making M and D. What we end up having to have happen is, in order for that B cell to start to make one of the other isotypes, we're going to have another event happen where the DNA gets changed. So we'll have another event known as class switch recombination. Remember, recombination is when we're going to break DNA and then repair it. And we're going to break the DNA and actually cut out these M and the D and this extra 55 kb and maybe even some other parts to allow the B cell to now start pairing its amazing VDJ region, the one that like got it pats on the head, the one that might even have just mutated to be a super all-star VDJ region, with a super ideal constant region. One of the ways that B cells know which constant region they should switch to has to do with which cytokines those B cells receive in the secondary lymphoid organ. So different cytokines made usually by helper T cells, but they could also be made by antigen-presenting cells, can induce B cells to class switch to make different types of antibodies. So you can see if a B cell receives IL-4, it might start making things like IgG1, IgG4, and IgE. If it gets IL-21 in particular, it makes some IgE. If it gets IL-10, it might switch more towards IgA. And so the particular cytokines, again, are going to influence the B cell's effector function, just like we saw happening with T cells. As I mentioned before, this process is a process of recombination, where we will actually have the DNA broken, and we will cut out some intervening DNA. So remember, M and D always come first, and then all of these other isotype constant regions come later. If, say, a B cell was going to switch to make IgG1, we would break the DNA here at the M site and at the G1 site. And that's what's being shown here on the lower right. And we would actually cut out that intervening DNA, and we'd now put the G1 constant region right next to the VDJ join. This is really happening because of these little sites. This figure is really, really, really not drawn to scale, called switch sites. They're little short sequences in front of each of our constant region parts of the DNA. And so we can have recombination events happen between the switch sites. So you can see you kind of get this like mixed switch site, and we throw out the intervening DNA. All of these details about class switching and affinity maturation and memory B cell development and plasma cell development, all of which are the big things we're looking for when we turn on a B cell, all of those things require B cells to interact with T cells so that CD40 on the B cell gets ligated by CD40 ligand on the T cell. There are some patients who are unable to have their T cells and B cells talk to each other. Those patients have a disease called hyper IgM syndrome. On the left, you can see a lymph node from a normal individual, and you can see that there are a bunch of germinal centers. If I looked at any of your lymph nodes right now, I would probably find some germinal centers because you're probably responding to some antigen. Perhaps you're actually defeating it and feeling totally fine, but you're making some germinal centers. If, however, a patient doesn't have CD40 on their B cells, or if that patient doesn't have CD40 ligand on their T cells, we can't see T cells and B cells interacting with one another. And so we can't actually see this process of germinal centers being created. So you can see there are no germinal centers in this patient. And in this patient, we won't ever see class switch. So all the B cells will make IgM, and they'll never switch to any other type. That, of course, means they're going to make too much IgM, but the really bad part is they're making not enough of all the other ones. So that's why it's called hyper IgM. These patients also don't do affinity maturation. They don't make good plasma cells. They don't have immune memory. And so this B cell T cell interaction is very important for making an appropriate B cell response. And you can see, again, those details and those results of the germinal center reaction here. So all of these things give us the important activation parts of B cells, of antibody secretion at a high level when that B cell has become a plasma cell, or that B cell can live for a really long time as a memory cell, or that B cell can undergo class switching, or that B cell can undergo affinity maturation. And I just phrase that like these are 4 exclusive things. The B cell does one or the other, but in fact, B cells often will do multiple of these things. They don't necessarily have to choose between one or the other of them. Exactly. We'll get a little bit more into kind of the memory piece of this. There's a little bit of a choice there, but that's a problem for a later lecture. So you can see kind of just a little bit about this process of production of plasma versus memory cells here. What I really want you to sort of look at is that on the left, our B cell, before it's interacting with the T cell, is kind of drawn like a normal old B cell. But then it gets drawn sort of differently as either a memory cell or a plasma cell. And in particular, it gets drawn really differently as a plasma cell. You've seen cells under a microscope in many lectures in this class and in a number of other classes. And on the left, you can actually see a plasma cell. The plasma cell looks really different than pretty much any other cell type, because what you'll notice is that the plasma cell basically has a nucleus, and then almost the entire rest of the plasma cell is rough ER. Rough ER is really, really good for a cell to be able to make proteins to secrete. And this cell basically stops doing everything else other than secreting antibody proteins. And so it gets rid of all the other parts of the cell and just has rough ER to just become a total antibody factory. That's what's being drawn here. And so this cell actually it loses BCR on its surface. You're not going to signal to that cell anymore. All that cell is going to do is just become a hardcore antibody factory. So what you can notice, those plasma cells do not have surface immunoglobulin. They don't have B cell receptor. They don't have MHC class 2. They don't grow. They don't do more mutations later. They don't switch classes later. The only thing they do is make lots of antibodies. So that's how they are unique from a naive B cell, just like remember we saw CTLs being unique from CD8 T cells or things like that. So when I first learned about all of this stuff, this was sort of the place or the part that we learned, that all of these things happen perhaps by magic wand, hooray. Also, during the time when I was in graduate school, some scientists actually found out the more details about exactly how, in particular, class switch or isotype switch and affinity maturation happen. Remember that we kind of think of those two things as happening around the same time in a B cell's life. One of the earlier slides talked about them being at slightly different times. Again, that's a little bit of a controversial thing that we could think about later. But at least we can say they're both things that are happening in the germinal center. So that's kind of similar parts of the B cell's life. And what scientists found was that both of these two processes rely on a critical enzyme. And happily, for all of us, that critical enzyme is the same critical enzyme that is required for both isotype switching or class switching and somatic hypermutation, which leads to affinity maturation. So there's this one really important enzyme that gets turned on during the germinal center reaction to allow both of these processes to happen. This enzyme is called AID. AID stands for activation-induced cytidine deaminase. Later this week, you will hear about a process a totally different process that immunologists had already named before this point that has an abbreviation of AICD. So, even though this is called activation-induced cytidine deaminase, the abbreviation AICD was already taken, so this became known as AID. But one thing that you might note just from the name is that activation-induced cytidine deaminase is only able to do its job when it's in a cell that is activated. And in particular, it's an activated B cell in the germinal center. The job of this enzyme is to do a process known as cytidine deamination, because it is an enzyme called a cytidine deaminase. And so we need to think a little bit about what in the heck cytidine deamination is. On the left here, you can see an example of a molecule of deoxycytidine. Cytidine, or deoxycytidine, is the C in DNA. So this is the structure of that C nucleotide base in DNA. And AID is able to deaminate cytidine. That means it can remove an amine group. And so you can see in this image that the sort of before chemical has this NH2, this amine group, and the after has an oxygen in the same place. And otherwise, it's really the same compound. So we've removed this amine group and we have this double bonded oxygen here. When this happens, that changes our cytidine into uridine. Uridine is the U that you learned about when you learned about nucleic acid. And one of the really important things that you learned about U in nucleic acids, or uracil, uridine, is that it is only found in RNA, not in DNA. If you go online, you'll find a lot of memes involving something called Chemistry Cat, and Chemistry Cat often says that if he had to pick RNA or DNA, he would pick RNA because it has U in it. So U, or uridine, is only found in RNA. That's really key in molecular biology. Here we have this enzyme, AID, that is going to act on DNA in activated B cells and specifically turn some cytidines into uridines. That's bad news. There's not supposed to be U in DNA. And now we've just turned some C's into U's. And so we've had AID act on the DNA and turn some of the C's into U's in that B cell's DNA. Those U's have to get dealt with. Sometimes there's just replication and they don't get dealt with, and we're not going to really deal with that right now. Sometimes we just have replication and that gives us a point mutation. So what used to be a CG becomes a TA. And sometimes we do different types of repair because we recognize that U is a problem, and we do some DNA repair. These ways of dealing with the uracil in DNA, these different sort of issues of how the uracil is handled, are the ways that we get either somatic hypermutation or class switch recombination to occur. And so when we have affinity maturation, remember, we're changing the affinity of the antibody, but hopefully making it better, though it doesn't have to be. Only the B cells we kind of see at the end of this process that live are the ones that did it better, but there are some that did it worse and died. They are mutating their DNA through somatic hypermutation. Remember that is picking up some mutations in the variable region of the antibody encoding genes, heavy and light chains. Those are actually specifically even directed towards the CDR region. So we tend to mostly see those point mutations over time in CDR1, CDR2, and CDR3. And so you can see even over time, we'll pick up more and more of those mutations. Well, those mutations are all the result of AID specifically deaminating cytosines in the variable region. And those cytosines leading, those now uracils that were once cytidines, now becoming situations where we have a point mutation
And so we can imagine that that uracil might get resolved in a way that just changes the base pair, perhaps to a TA, perhaps to some other types of base pairs. And so we just might put in a random other base pair in order to deal with that uracil. And if that happens in the variable region, at one of the cytosines in the variable region, that will actually give us our somatic hypermutation that can lead to affinity maturation. We can also see AID acting at a different part of the DNA of the B cell. And here, that AID is going to act on cytosines in the switch regions, upstream of some of the different constant regions. Which switch region we act on will be based on cytokines that that B cell received earlier in its activation. As a result, that switch region will actually have a fair amount of damage to it. We'll have a lot of DNA damage because there'll be so many cytosines, and we'll end up having to go through sort of this kind of a DNA repair process with of the double strand break. And the way that this will work is that the cell will try to find a similar piece of DNA that it can do recombination with to repair that switch region. And oh my gosh, there are all of these other switch regions that are very similar DNA. They can actually come do recombination to repair the DNA. That will cut out the intervening DNA. So here you can see we had these AID initiated double strand breaks, and we're going to do recombination. It's really a version of homologous recombination to try to repair the switch region. That will eventually cut out the intervening DNA and move that new constant region close to the VDJ join. And so that's what's going to allow us to cut out this large amount of DNA in the switch region and resolve that uracil. So at the very end here, I'm going to show you a little video of the germinal center reaction process that I got from another scientist who works a lot on this process. So here we can see how this works. So first you can see a naive T cell getting activated to become a T follicular helper cell. Oh my gosh, it became a T follicular helper cell that makes that transcription factor, BCL6. It's making its cytokines, it's making CD40 ligand. It goes to the area where it can interact with B cells. And now we have that B cell starting to become activated. It had been activated a bit by antigen before. So now that B cell is going to first have been activated in the light zone. Now it moves to the dark zone and proliferates and does hypermutation. And then it can go back into the light zone, test again to see if it did a good job, and eventually leave as a memory or a plasma cell. Eventually the T follicular helper cell will also be a memory T follicular helper cell. And so you can see kind of the same process happening here. Here's our activated T cell like we saw before. It could become like a Th2 cell and go do other things, but we don't care about that today. Or it could become a T follicular helper cell. Here's our B cell that comes into this area. It maybe gets activated. It hopefully interacts with a T cell, then maybe goes to a germinal center, tries to interact with T follicular helper cells and follicular dendritic cells multiple more times, turns on AID as it's doing its mutation and potentially class switches, and may leave as a memory cell or a plasma cell. This process is incredibly important in understanding B cell activation. It's also incredibly important in understanding some details of how we make vaccines, or even how we try to make antibodies in the laboratory, because we need to know how to turn on a B cell really well. And so we'll see a lot of ramifications of this later on. Thanks for putting up with a video lecture, and I hope you guys are having some good days. I'll talk to you later.